So good day everyone, I'm Johan A. Francisco from BSN Industry, and today we are tasked to do the experiment number 9 for the DNA extraction. So let's start. So as you can see, we have here our materials for our experiment. We have here our beaker, scoop, sodium chloride, liquid dish soap, paper cup, alcohol, improvised test tube, improvised dropper, gloves, and bottled water. So, hey there guys, I am John again. So we have here our materials for our experiment. So first is we're gonna prepare a 2 large scoops of salt and also the 25 ml of distilled water. So we're gonna create a saline solution in a beaker by adding 2 large scoops of salt to approximately 25 ml of distilled water. And then after this, we're gonna get a spoon to stir until the salt is completely dissolved in the beaker. And now it's done, we're gonna pour the salt water into the paper cup slowly. So after stirring it, we're gonna drink a mouthful of the solution from the paper cup and switch it back and forth for at least 30 seconds, occasionally scraping your teeth along the inside of your cheeks as you do. It's the best to do this with a clean mouth, not right after the lunch. And then we're gonna spit our mouthwash solution back into the cup. And then we're gonna bend the cup into a sort of spout and pour the mouthwash solution into the test tube until it fills about 1/2 inch of the bottom of the test tube. After this, we're gonna carefully add 2 drops of the liquid soap. Since the bottle of the alcohol cannot be opened, we're gonna pour an ample amount of alcohol into the paper cup. And then we're gonna get our improvised dropper and put an ample amount of alcohol into it. And then we're gonna tilt the test tube approximately 45 degrees and add 20 drops of the chilled alcohol so it slides down the test tube without disturbing the solution. Since it's less dense, the alcohol will sit atop the mouthwash and soap solution. And now we're done adding 20 drops of the chilled alcohol into the solution. Assuming that this is the cup, we're gonna tightly put the cup on the test tube and very slowly and gently tilt it upside down, then right side up 3 times. Do it carefully so as not to make bubbles. After tightly putting the cup, we're gonna wait for about 1 minute. After 1 minute letting the test tube sit undisturbed in an upright position, at this point, you should begin to see a milky white thread, possibly interspersed with bubbles, appear between the solution and the alcohol. And as you can see, we have here, and that is our DNA. After several minutes, the DNA should be suspended in the alcohol layer. After seeing our DNA extractions, we're done now doing the experiment. Thank you and goodbye.
Guide question number 1. What is the purpose of soap in the experiment? The soap breaks down the lipids or the fats and the phospholipid bilayers of the cell membrane and nuclear membrane. This releases the contents from the cell and the chromosomes containing DNA from the nucleus. In short, the purpose of the soap is to help break down cell membranes and release DNA.
Guide question number 2. What is the purpose of using ice-cold alcohol? First, its use can be that as the temperature decreases, there is more and more precipitation. Second, using ice-cold ethanol can be that the enzyme DNases, like other enzymes, are temperature sensitive and therefore DNases are not active when the temperature is too low. Since these enzymes are involved in DNA lysis and degradation, reduction in this enzyme's activity will yield more amount of pure DNA. Lastly, DNA contains the blueprint for the development of functions in all living organisms. The instructions are embedded along the strand of DNA, or what we call genes, which is responsible for the production of proteins that determines the type and function of a cell. From our past experiment, we have learned that there are different factors which contributes to the denaturation of protein, which includes heat, strong acids, and organic solvents. Hence, the purpose of using ice-cold alcohol is to avoid the breakdown or denaturation of the DNA. Thus, DNA is water-soluble. Using an alcohol renders it insoluble. The positively charged sodium ions and negatively charged DNA phosphate groups combine to generate strong ionic bonds, causing the DNA to precipitate, resulting in a clumps of visible white strands. In conclusion, using an ice-cold alcohol is critical in order to extract a higher amount of DNA.
For guide question number 3, where can the error be in getting only a few threads of DNA in the experiment? For our answer, banana plant is known to have high amount of polyphenols and polysaccharides, which interfere during usual DNA extraction process and usually remain as impurities in the DNA template preparations. Moreover, the ripe and overripe bananas produced a smaller amount of DNA because the cells that stick the nutrition break down and begin to decompose when it begins to ripen.
For the generalization, so this experiment was a lot of fun to do since we learned new things and had new experiences. We were first interested in seeing how DNA would look like in a basic experiment. But then, after completing the experiment, we were astounded by the actual result. We began our experiment by gathering all of the necessary supplies for extracting DNA, such as container, stirring the saline solution, and many more. To extract DNA from our cheeks, we swished salt water about in our mouths and scraped the insides of our cheeks with our teeth. This has allowed us to gather a sufficient number of cheek cells. Because of the mixing of soap and mouthwash solution, as well as the influence of chilled alcohol on the sample, DNA strand was formed.